Welcome this afternoon. After lunch, the third session. This morning, the focus was on membrane technology in water treatment. This afternoon, the focus is on biological processes in water treatment. And the first session will be chaired by Kees Buisman. Kees Buisman is professor at Wageningen University and he's scientific director at WETSES. Kees, the floor is yours.
Well, good afternoon, ladies and gentlemen. We tried to make it a very inspiring session with magical stories and brilliant discussions, and especially that everybody keeps himself and herself to the time. So I will not spoil any more time, and I would like to introduce the first speaker. It will be Mr. De Vet from the Netherlands, and he works at OAS, at the drinking water company, looking at the biological system for drinking water production. Please, take the floor.
Thank you very much, Kees, for your introduction, and good afternoon, everyone. Well, my name is Wieren De Vet from the Drinking Water Company. Well, I should first start, I guess. That's already a challenge. You do that for me? Right. Now, 20 minutes from now. My name is Wieren De Vet. I wrote a paper for this on this topic for this conference with my colleague and co-author Willem-Jan Knibbe, but unfortunately he has been struck by bad bugs. He has the flu. He is at home at this moment. Yeah, well, it's not related. It's just in the air, I would say. But I'm going to talk about the good bugs in drinking water treatment. I would also like to acknowledge Luc Rietveld and Mark van Loosrecht from the Delft University of Technology, who have been working with us on this topic. Biological filtration is, of course, a very wide notice. You can think of anoxic processes to remove nitrate and perchlorate from water. You can think of all kinds of processes removing organic micropollutants or natural organic matter. And of course you can think of other processes, stabilizing water, for instance. But today I'm going to talk about aerobic filtration, so filtration under aerobic conditions, and then aimed at the treatment of groundwater, anaerobic groundwater. To remove iron and manganese, methane and ammonium. I should point there. Where should I point? Okay. I must excuse for this slide because you cannot read it, so I will talk you through it. I wasn't able to change, I didn't have time for it, but I mention here the four macro groundwater compounds: methane, iron, ammonium and manganese, with the concentrations we find at the drinking water company of Oasen. That is up to over 10 ppm for methane, iron and ammonium, and 1 ppm for manganese. You can remove these substances, of course, by chemical oxidation with a strong oxidizer. Excuse me. Or you can use separation techniques like membrane filtration or stripping. But when you take away the strong oxidizers, then you will see that all these compounds can also be oxidized just with oxygen from the air by microbes. You cannot read it, and you do not have to read it, but each of these compounds can be oxidized by specific groups of microorganisms. Well, when you think of natural biological filtration, it's actually quite close to a natural system where you have rainwater or surface water infiltrating in the ground. To the left side you see, I would say, engineered infiltration system, a filter. At the drinking water company of Oasen, we often use dry unsaturated trickling filters. So you don't see much water on top of it, it's just being sprayed and the filter is dry. We do this because we have high concentrations of ammonia and also methane, and we don't want to have anaerobic conditions in these filters. Well, the scheme is, well, when you look at that this way, it's quite simple. You spray the raw water with methane, iron, manganese, ammonium on top. You also bring air from oxygen with air at the top. And then you see in the filter, you get conversion from ammonium to nitrates, and you get precipitation of iron and manganese. And then if everything goes all right, you can see that the methane leaves the filter by the air. Well, I'll let you look at this for a while, and then when you understand it, I will go on. Excuse me. I also had a cold. When you talk about biological filtration, there's often a problem that people say, well, how important is it? Nowadays, luckily, we can use a lot of molecular DNA techniques to get more insight in this. So to see what kind of bacteria or microorganisms are present, and also in what numbers they are present. So these are actually just gene codes. At the Oase, we used some of the techniques to characterize the system, so the groundwater and the filters. We used DGGE to find the composition of the populations in filters and in groundwater. And nowadays, of course, you have newer techniques that are better and more, I would say, give more results, like new generation sequencing and clone libraries. But we used this, and it already gave a good impression of the populations living in these groundwater filters. And of course, when you have nitrification, you find bacteria like Nitrosomonas in the filters. But we also found archaea in the filters, ammonium oxidizing archaea. And we found Nitrospira bacteria to oxidize nitrites. So it's not Nitrobacter. And Nitrobacter you find in systems with higher ammonia and nitrite concentrations. But in these systems with low concentration, you find Nitrospira bacteria. Well, for the other metabolic groups, iron and manganese oxidizing bacteria you also found. So this is a typical iron oxidizing bacteria, and this is a facultative manganese oxidizing bacteria. We also found in the groundwater and the filter systems. And then in the end, with this technique, we did not find any methane oxidizing bacteria. But I will come to that later. This takes some time. Well, another, and I would say a more sensitive technique to characterize these systems is quantitative PCR, where you can actually really indeed look for specific species in your filters, and then, well, go to actually one cell in a milliliter or less if you want. So really very sensitive method. We use this technique to quantify ammonia oxidizers, methane oxidizers, and iron oxidizing Gallionella bacteria. And then when you have these results, you can schematize the filter, and then you can measure the concentrations of these bacteria in the influent, effluent, and the backwash water. And with these numbers and the flows, of course, you can make balances of how these organisms grow in the filters. Now, for the first process, iron oxidation, when people talk about biological iron oxidation, the first thing they think of is some sort of a layer between an anaerobic anoxic zone and an aerobic zone. So where anaerobic groundwater comes to the surface, you often see in nature the growth of iron oxidizing bacteria. You can make this into an engineered system. Yeah. So this is a picture from our Flemish colleague company of Pitpa. And it shows actually the same sort of system, with the groundwater coming here and then streaming very quickly on top of a filter. And you also get mostly biological iron oxidation in these filters. Well, I already talked about the Gallionella. One of the main, I would say, specific iron oxidizing bacteria is the Gallionella bacteria. And this bacteria can actually be characterized through DNA, but you can also see it in the sludge. When you backwash a filter or you look into the filter effluent, you can actually see these deposits called twisted stalks. And these are not the bacteria, these are the bacteria, but these are just iron deposits, but in a very characteristic form, so you can really see if they grow. And the nice thing from this bacteria is that they can only grow on iron oxidation. So if they grow, you know that there is biological iron oxidation. Sorry. Now for engineered systems, first a system that's really close to, I would say, natural iron, biological iron oxidation. That's in situ iron oxidation. This is a system where you every now and then infiltrate a little bit of aerated water into the anaerobic groundwater. And here you can see a pattern of iron on the vertical axis in time coming out of that particular well. And you see cycles, so you see that the iron concentration goes up, and then after you infiltrate aerated water, it goes down and comes up again. So this is one cycle of in situ iron removal. And we looked at if Gallionella bacteria were actually growing this well, and we found these results. This is one cycle of this in situ iron removal, so 42 days in this case. And on the vertical axis you first see some dots here, which is on the logarithmic scale about 10 to 100 cells per milliliter. And then here on the vertical axis in log scales, this is 10 to the 5th and this is 10 to the 7th. So you see that after this observed in situ iron removal, you get a really huge increase in Gallionella cells coming from this well. Here you see the pictures. It's not very clearly here, but you can see the same twisted stalk deposits again.
Where do you see this increase? I see a decrease.
Yeah, but you should compare it to this one. So this is the cycle. So at the beginning of the cycle you have more cells coming out. And at the end, well, it's still a factor 100 higher or 1,000 higher. Now I go to a more extreme system, trickling filtration. So the general perspective is that these Gallionella, they really grow under low oxygen conditions. In trickling filtrations, you aerate a lot, and the water coming out is oxygen saturated, so it has really high oxygen content. The pH is about 7.4 in these cases. So, well, in the first glimpse, you would not expect much of biological iron oxidation. Still, we are doing these tests now for half a year. You see they work very well. So we have two filters running at this time with filtration rates from 10 and 30 meters per hour. Excuse me. And you see that of the 6 milligrams per liter ppm of iron coming in, well, almost everything is being removed. So they work really well. And you see when you look at the filter run pattern, that is, well, there's some elevation at the beginning, but it's really quite good. The max is 0.5. And so they work well, but is it biological or not? That's the question, of course. Well, this is the result of one of these balance calculations, where we make a cumulative calculation of all the bacteria coming in the filter during in the influent during one filter run time. Well, this is quite a big number. But then when you go to, if it works, when you go to the effluent and the backwash water, so also accumulated over one filter run time, you see that it is, well, again a factor 100 to a factor 1,000 higher than the number of Gallionella coming in. So you see a very clear increase in Gallionella in these filters. And when you relate it to literature, you can indicate that about, well, quarter to half of the iron oxidation has been done biologically in these filters. Excuse me. Now the same for, if you want, for methane oxidation, because methane can of course also be very easily oxidized by microorganisms. but it can also be stripped. So we did the same measurements for methane oxidizing bacteria. And then you see these results. You don't see anything? Yeah. So you can actually see that the number of cells leaving the filter are, well, actually even a bit lower. But this is the detection limit of for the method for the influent concentration. But you can clearly see that there's no big growth of methane oxidizing bacteria in these filters. And from this we conclude that methane oxidation is mainly a physical, it's not happening, and it's mainly a physical stripping process taking place in these filters. We are confirming this with measurements from methane in air measurement and so on. Well, the last thing I want to bring to your attention, sorry. Of course, when you look at these groundwater filters, they are a very complex system. So you have several processes. There may be methane oxidation, at least biological iron oxidation, nitrification, maybe manganese oxidation. And in this graph, I show you the effluent quality of one filter for ammonia during the period of half a year, which is really combining these processes. So the iron and ammonium removal are in one filter. These filters operate at 2 meters per hour, so rather low filtration rate. Influent concentration is about 5.3. And you see this effluent, well, it is lower, but the removal is only 60 to 80%. And then when you separate the process, so you use this quick iron removal step before this filter, you see this pattern. So you also have a startup period for the nitrification, but then you have almost complete ammonium removal. So we come to the conclusion that there is some sort of a bad influence from the iron on the ammonium removal. We do not exactly know how it works. There are strong indications that it works through nutrient limitation. And we think mostly of phosphate as a limiting nutrient. So I will not go into this now, maybe later in discussion if you're interested. But here actually we show that without any phosphorus dosing, we have no nitrification. So there is no nitrite and nitrate being formed. And when you dose phosphorus to this water, you can see that nitrification starts. So there's a clear indication that this is the limiting nutrient in these systems. I can tell a lot more, but I have limited time, so.
4 minutes.
Okay, well then we can not discuss. But I come to my final slide. And, well, I'm in time, I think.
We will do the discussion later, but is there maybe a clarification question? The urgent one? Then, thank you very much.